Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Unlike plants, animals can't carry out photosynthesis to generate our own oxygen. Yet our brains rely on oxygen to make the massive amounts of energy needed to function. In a study published today, October 13, in iScience, researchers found a way to harness photosynthesis to supply neurons with oxygen. They injected either cyanobacteria or green algae into Xenopus laevis tadpoles and deprived the animals of oxygen, causing brain activity to cease. Exposing the animals to light, which allowed the microbes to make oxygen from CO2, restored neural activity. The authors employ an elegant and easily reproducible experimental approach to examine the effects of activation of photosynthetic organisms as a way to directly increase oxygen levels in the brain. Diana Martinez, a neuroscientist at Rowan University in New Jersey who was not involved in the stew. Over lunch, Straka and LMU plant biologist Jorg Nicholson got to talking about how they might work together on a project. Their solution: investigate whether it would be possible to have photosynthetic microorganisms supply the brain with oxygen. But they were able to restart activity in the brain by shining light on animals who'd received injections of microorganisms. When they turned off the light, neuronal activity ceased again. Although the experiment was a success, Martinez notes it's not clear whether the findings could be translated to treat conditions in which the brain is starved for oxygen. The first issue is that Xenopus laevis tadpoles are transparent and light can easily pass through the skin to activate photosynthetic machinery to produce oxygen. Use in more complex animals would be difficult, as light does not easily traverse the skin and may not reach the vasculature to activate the photosynthetic organisms, she writes. Additionally, while low oxygen can be a problem, excess oxygen can also exacerbate brain injuries. Thus, the inability of oxygen levels to be controlled properly through the use of these photosynthetic organisms would therefore be just as detrimental as the hypoxia itself. Trying the technique in brain organoids and slices first would give more insight into its physiological effects, she adds. Over the last decade, there are quite a few projects where people have been trying to set up artificial symbiotic associations with algae, in order to augment in some way or manipulate vertebrate physiology, which is really radical, says Ryan Kearney, a biologist who studies symbioses between algae. Approaches where microbes are artificially inserted into cells or into tissues to modify their function are largely unregulated and under-scrutinized in comparison to widely used genetic modification techniques such as CRISPR that target one gene, Kearney adds. The unknowns, as well as examples of pathogenic algae, make this strategy a bit risky, he notes. But the potential implications are also just fascinating to speculate about. Can we get away from breathing as a way to keep our brains going? Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.